Hello, dear students. In last lecture, we have discussed about the standard plate count method. It is a qualitative method for the detection of determining the bacteriological quality of drinking water. Now, in this lecture, we will discuss about the MPN method for determining the quality of drinking water, whether that water is potable or not. This method is devised to detect the presence or absence of coliforms in water sample. It is somewhat a lengthy procedure. In this method, the different selective and differential media are used for the detection of water sample. This test is carried out in three different steps, that is presumptive test, confirmatory test, and complementary test. So let's start with the test. What is meant by presumptive test? In this test, that is presumptive test, the water sample which we have collected, we have to properly mix the water sample which has we have collected earlier. The bottle of the water to be tested properly mixed. After that, we have to inoculate 5 double strength laurel double strength lactose broth, or we have to already prepared 5 double strength lactose broth tube, or the other are single strength lactose broth tube, or we can other in or at the place of lactose broth we can also use laurel tryptose broth on the place of lactose broth. So we have to inoculate 5 double strength lactose broth tube. In that tube, which contain double strength lactose broth, we have to add 10 ml of water sample. In another 5 ml single strength tube, which are already prepared, in that we have to add 1 ml of water sample, and 5 tube of again single strength lactose broth, in which we have to add zero point 0.1 ml of water sample. So here we have to inoculate total 15 tubes, 5 of double strength and 10 are for single strength lactose broth. In double strength, we have to add 10 ml of water sample. In 5 ml single strength, we have to add 1 ml, and another 5 single strength, we have to add 0.1 ml of water sample. And all these tubes are incubated for 24 hours at 35 degrees Celsius. After 24 hours, we have to observe for the formation of acid and gas. Acid is also detected by the color change of that media. It becomes yellow in color, and gas formation is observed by means of formation of gas bubble in the inverted Durance tube. The tube which shows the acid and gas positive, that tube is considered to be positive for the presumptive test. Sometimes only acid formation are observed, while gas formation are not observed. Uh, so that tube are considered to be the negative for in case of presumptive test. After that, after 24 hour or 48 hour, we have to proceed for the confirmatory test. From the tube which shows higher dilution and shows gas formation, including acid formation, we have to transfer one loopful of culture to the BGLB broth, and we have to incubate that tube for 48 hours at 35 degrees Celsius, and we have to also observe for the formation of gas within that incubation period. And if the gas formation can observe, we have to consider that tube as a positive for the confirmatory test. Here, many times only acid formation can be observed, but if there is no gas formation, that tube is considered to be negative for that particular test. After, we have to proceed for the completed test. In this diagram, we can see it is nothing but the water sample which we have collected from the particular water source. We have to already prepare single strain and double strain lactose broth. In double strain lactose broth, we have to add 10 ml of water sample in each tube. In this tube, already we have inserted an inverted Durance tube. You can see in this diagram. After that, in another 5 tube, which contains single strain lactose broth, we have to add 1 ml of water sample, and another next 5 tubes, we have to add 0.1 ml of water sample, which we have collected earlier. We have to incubate for 37 degrees Celsius for 24 hours, and after incubation, we have to observe for the formation of acid and gas. In the diagram, the color change we can observe. That means it is nothing but the yellow color, and the gas formation, so that the tube is considered to be positive for the presumptive test. If out of these five tube, only two tests are considered to be positive, as it shows acid and gas formation. And in case of third tube, we can see there is a complete tubes are there is no acid and gas formation. Gas formation we can observe like this. So these are nothing but in case of presumptive test and PGLB broth, when we use this look like this type. So after the presumptive test, we have proceed for the complete confirmatory test, and after that we have to proceed for the completed test. We have to uh, inoculate the strict loopful culture from a positive BGLP broth, which we have already incubate in the incubator for 24 hours. And from that positive acid and gas forming tube, we have to inoculate loopful of culture on EMB agar plate, and again that plate is incubated for 37 degrees Celsius at 24 sorry 37 degrees Celsius for 24 hours, and we have to observe the colony characteristics of that particular organism, which can be shown on the eosine methylene blue agar or EMB agar plate. So here the characteristic pattern of that organism which shows growth on that particular tube, we have to observe. In case of the coliform which are already present in that water sample, they are from the fecal origin, or the organism which we have already considered as an indicator organism, that is nothing but the E. coli from fecal origin, and enterobacterial origins from the non-fecal origins. So E. coli shows a typical type of colonies, which are nothing but the small in shape, dark center, and greenish metallic sheen can be observed for the E. coli type of organism. And here we can confirm or complete our test by means of that sample is contaminated with the fecal coliforms. And in case of organisms are grown on the EMB agar plate, but it shows atypical colonies like large pink or colorless colonies, non-nucleated, without greenish metallic sheen. These are nothing but the differentiating characteristic between fecal and non-fecal coliforms. On that basis, we can confirm that that water sample is not contaminated with the fecal matter. Presence of typical as well as atypical colonies constitute a positive completed test. Typical colonies are selected preferably, while in absence, atypical colonies are selected from EMB agar plate and inoculated into lactose broth, that where coliform produce gas, and on agar slam they are gram negative and non-sporulating bacilli. So after the growth on EMB agar, we can also proceed for their morphological characteristics. So here we can observe whether they are gram positive or gram negative spore former or not. On that basis, we can confirm the uh, that um, um, that organism which from fecal or non-fecal origin. Here in this diagram, we can see these are the nothing but the EMB agar plate, and here these are nothing but the dark center, small type of metallic sheen type of colony, and these are nothing but the characteristics pattern of fecal coliforms. And here we can see a pinkish or colorless colonies of non-fecal coliforms on EMB agar place. So on that characteristics pattern, we can differentiate that our water sample is contaminated from the fecal matter or not. Here, the fermentation of lactose broth and demonstration of gram-negative non-spuriating bacilli constitute a positive complete test. These are nothing but the characteristics pattern. For separation of coliform into fecal and non-fecal coliform organisms, a combination of immune tests are performed. So immune test at the same time we can inoculate all the four tube, and after 24 hours we can get the result. So it is nothing but the duration of the test is nothing but 24 hours we can take. For this MPN method, we can use at least 72 hours will be required for the completion of the test. So it is quite lengthy procedure. Presence of fecal coliform organism in water sample shows evidence of fecal pollution, indicating that intestinal pathogenic microorganisms may be present. It is nothing but the indicator. Thus, water source is not safe for the drinking purpose. So, on that result, we can confirm that that water sample is contaminated with the fecal matter, and it is not potable for the human consumptions. Here, in case of MPN table, we can also estimate the most probable number count in that particular tube. So here MPN index we can use. MPN for 100 ml is equal to nothing but the number of positive tube, which is nothing but the considered for the formation of acid and gas for the tube. So total number of tube which shows acid and gas formation multiplied by 100, divided by underscore ml of sample in negative tube multiplied by ml of sample in all tube. So by counting the volume, we can get a MPN index of that particular water sample. So in that table, we can also see these are nothing but the number of tubes giving positive reaction, out of which 5 undiluted samples. Here another column, second one, and third we have already prepared 3 sets of different tubes. So if we can observe 1 tube from that sample, 2 tubes from the what sample, and 0 tube from the third gate, we can get nothing but the six nothing but the MPN index of that particular water sample. So by adding that particular value, we can get a MPN of that most probable number count of that particular water sample. So here nothing but the table we can use normally for the MPN calculation. Thank you.